for the study, and he is currently a graduate student at Washington University. Laura Iannotti is unfortunately unable to be on the webinar, and she was a PI of the study. So, as okay, do I do this here? Should be fine. Oh, no wonder. Yeah. Okay, starting by, okay. So, as Emanuela said, that the prevalence of, or the rates of stunting are a barrier, not only to individual development, but also national development. And the sub-regional prevalences that we see here in this slide, that look fairly low in South America and Central America relative to Africa, certainly, and Southern Asia, really high, large disparities among countries in the region as well as within countries. For example, Guatemala, 1 in 2 children are stunted, and the prevalence really hasn't come down very much. Other really hard-hit ethnic groups in the region are those of indigenous descent. And this really affects the Andean countries primarily, Bolivia, Ecuador, and Peru. So trends are, the numbers of stunted children are going down in Asia and Latin America and the Caribbean, rising in Africa. In Africa, there's been less decline in prevalence. Population of young children is also going up. And currently, there's over 150 million stunted children globally. Also, following up on Emanuela's introduction, stunting is seen as a key barrier to economic development. So in this photo, you can see the young girl on the left is actually older than the girl on the right, but you wouldn't know that she's stunted unless you measured her length or height, and then you could compare it to her age. And to quote your president in the annual meeting last year, he said, the effects of stunting have lifelong consequences, not only for the individual, but for countries as well. How will countries compete in a global economy in the future when 30 to 45% of their children do not have as many neuronal connections in their brain? So absolutely drawing the link between stunting and early child development, and we'll certainly touch on that in today's presentation. So in general, and certainly in Latin America, where I spent 19 years of my career, complementary feeding foods as well as practices are still a barrier to child growth and improved nutrition. So what we're seeing in the 2 photos on top are really, very, very thin broth. In Peru, they think there that the broth is the essence of the food, as thin as it is, with a few little pieces of starchy white rice floating around. This picture from Colombia, you can look on the plate, starch. There's like 1 or 2 little beans there. A child's not going to grow well with this kind of food. Another big issue still in the region is lack of attention to what kids are eating. They're just kind of, given some food, set out there, and so this child has to worry about, are the chicken's going to eat his food, who's interacting with this child. Important point that it's not just about what you eat, but how the child is fed. This is a time of interaction. Talk to the child, love the child, face the child. It's child development too. It's not just the food. It's also the feeding practice. To date, there's been 2 recent systematic reviews, meta-analyses that look broadly at what's going on with complementary feeding interventions in the Maternal and Child Lancet series in 2013. Zubi Boussa and his colleagues did a review, and they said, the provision of complementary foods in insecure populations was associated with significant gains in height for age Z-score, standard mean difference of about 0.4. That's a good size effect. But more recently, just published, 2 investigators at Johns Hopkins, another systematic review and meta-analysis, and this was really to contribute to LIST, to be used in LIST analyses, they concluded that complementary food supplementation interventions with or without nutrition education had a small significant effect in food insecure settings on length for age, but they only got less than 0.1 standard mean difference. So that's kind of where we are with the state of the art in terms of the systematic reviews. Why did we get interested in eggs? Christine, Laura, and I call ourselves the egg team. You know, we are committed to this. And I'm thinking around this when we really started talking at length in 2013 was that, you know, the focus of research for complementary food feeding has really shifted to products, which are useful. I'm not saying they're not useful. I'm saying that in certain settings they are useful. I think there's questions of sustainability, scalability. We have all worked on these foods. You know, I was involved in this program in Ecuador, Fondos Mill, year 2000, national program of fortified complementary foods. Laura and Christine have definitely worked on the lipid-based nutrient spread. But our feeling was we have to broaden the research agenda. We also have to look at food-based approaches. And we just felt we were getting too narrow in terms of how the research was going. So why did we become interested in eggs? There was limited data, published literature that showed an association of eggs and growth, but no randomized controlled trials, nothing that would say this was a causal effect. Eggs are also the most inexpensive source of animal source foods, and we're really recognizing animal source foods are absolutely key to a child's development. So how do we get money for research? Well, you publish a paper and you give it a catchy title. People really remember this title. And we start by saying eggs have been consumed throughout human history, though the full potential of this nutritionally complete food has yet to be realized in many resource-poor settings. We collaborated also with a vet because we really felt that we had to look at the animal poultry piece of it as well as the nutritional piece of it. Again, thinking that it's got to be sustainable, it's got to be scalable if it's effective. And then we also looked at during pregnancy and lactation. That's our next big push to get funding. Do you know anybody? And it starts with, it should not be surprising that a simple egg, which provides a chicken embryo nutritive support from conception to the time it hatches, might also be important food to support fetal growth and development during pregnancy. So this is our paper from Nutrition Reviews, and basically what it shows is that eggs contribute a large portion of key nutrients for a child that's breastfed between 7, we just picked 7 to 12 months, but we could have picked other age ranges as well. More than 50% of protein and other micronutrients associated with cognitive development, and Christine will definitely touch on this, but also significant contribution to energy intake as well as other very important vitamins and minerals. But eggs are much more than nutrition because this whole concept of a food matrix is more than the sum of its nutrients. You know, a food, an egg, is a biologically complex. It consists of cells, other non-cellular materials, and their molecular constituents. And eggs, as well as some other foods, are complex, non-random mixtures of natural compounds developed under biological and evolutionary control. I mean, eggs go back millennia. And just an example, you know, the yolk of an egg is the nutritive part, while as the white really provides immune protection, stability, you know, to this growing chick. So the Loon. The Loon Project was a collaborative effort. Laura, as I mentioned, was a PI from Washington University. We had Universidad San Francisco de Quito in Ecuador. I was at the Pan American Health Organization, Christine from UC Davis. And we're very, very grateful to the McKeel Institute that really took a risk on us and provided a small amount of seed funding to really look at this proof of concept idea. Specifically in Ecuador, you know, similar to what we saw in my earlier slide, stunting has decreased over time. 1 in 4 kids are still stunted in Ecuador, but it really varies by ethnic group. So what we're seeing in populations that identify themselves as indigenous, over 40% of children suffer from stunting. So what did we set out to do? We set out to test the efficacy of introducing eggs early in complementary feeding on growth and biomarkers. Our primary outcome were biomarkers of choline, betaine, vitamin B12, fatty acids. So these are nutrients associated with cognitive development. And of course, we were interested in anthropometry and growth. In addition, we were really interested in acceptability. Would mothers give these to their kids? Dietary intakes, morbidity, amino acids, as well as different growth factors that, in addition to these nutrients, really are associated with improved growth. We had approval from 3 IRBs to conduct this study. The design was a randomized controlled trial in a couple hours outside of Quito in Cotopaxi province. It's a mixed indigenous community with a very high baseline stunting among the young children. The intervention was very simple. We gave 1 egg per day for 6 months, and the eggs were purchased locally. They were delivered weekly to the families for their young child. We recruited children very young, between 6 and 9 months of age, a period when growth is undergoing really rapid change. And we collected M-line data 6 months later when these kids were 12 to 15 months of age. We used mixed methods. We really started with qualitative research that was led by Will Walters, very much grounded in theory, structured observations, focus groups, in-depth interviews. Extensive quantitative data, caregiver surveys, anthropometry, and then, of course, the biomarkers, some of which were analyzed in Ecuador and some at WashU. I'd like to take a minute to really talk about the social marketing component of the study. It was based on the qualitative research. It was, I think, unique to the study, and Carlos Andreas, who's on the phone, really led this effort. What it intended to do was create demand by overcoming social, cultural taboos that would prevent a mother caregiver from giving eggs to her child. And we branded it as the Loon, which translates to egg in the local indigenous language, or Quechua. And what this did is symbolically really tied the practice of giving eggs to indigenous world views. And as you can see, the egg is wrapped in a Quechua shawl. You know, the colors, it was really, and then the figure on the left is really a typical Ecuadorian figure, very well known. It looks scary, but kids, everybody remembers this figure. And when we recruited and had to draw blood and do all this other stuff, you know, this guy was dancing around and keeping the kids entertained. And so, you know, the community really associated branded project and also the cultural significance of pulling in this particular individual. It created brand loyalty. It helped empower the mothers to take decisions, and it was really critical to the success of the study. So it was a relatively small sample size. We really didn't have a lot of money. We randomized 163 children to interventions or controls, and we had 83 in our control group, 80 in our intervention group. And at the end, we had only lost 7%, which is, again, I think a huge tribute to the social marketing, because a nearby volcano that hadn't exploded in years decided to explode. Ash was everywhere, and a lot of people left the area for a while. So the growth effects, just to focus on, that's the first paper published in Pediatrics in June of this year. Just to focus, we had an effect size of 0.63 length for age Z-score, highly significant, and this translated also into a 47% reduction in stunting in this population. So if you think back to what these meta-analyses, systematic reviews were showing earlier, a lot of people say, well, it can't be true. So, so we'll see. We think it is, but you know, obviously we've got to keep doing our proof of concept, and Christine will talk about that. And I think another really important point in all of this is that the whole distribution of the intervention group shifted to the right. There was no threshold. Every child that got an egg really benefited from this intervention. So I am going to turn it over to Christine to finish the results and also talk about our next steps and conclusions.
Okay, thank you, Chessa. So I'm going to be talking a bit about the nutrient biomarker analyses that we've done with a focus on some of the work that we've done to try to look at which pathways, which nutrient pathways may be partially explaining some of the growth effects that we've seen. So when many people think about eggs, they think about the protein. Eggs are considered to be a complete protein and a very high quality protein. And when you look at the amino acid composition of the eggs, you can see that they're very well matched to the amino acid requirements, especially of infants. And we did the nutrient composition of our Loon eggs, and so that's what I've shown here on this slide, and then matched them against the amino acid requirements for the infants at baseline and at end line. And for infant and child amino acid requirements, they're expressed on a milligrams per kilogram of body size. So as the child grows, their requirements are getting higher over time. So that's why you can see at baseline, when the infants were about 6 to 9 months of age, for many of these amino acids, an egg a day was meeting 100% of their amino acid requirements. As they got a little bit older, it was a little less, but nevertheless, it was still meeting at least 50% of the amino acid requirements, and in many cases, 80 to 90% of the amino acid requirements. But we really think that eggs are more than just the amino acid and the protein. And in fact, when you look at the other nutrients, the vitamin and mineral composition of the eggs, they are a very rich nutrient source beyond amino acids. And in particular, what I want to draw your attention to is the choline concentrate, the choline content. Choline is a conditionally essential nutrient. The requirements are higher for pregnant women and young children. Choline is involved in fetal and infant brain development, and eggs are really the most choline-rich source of food in our diets. And for infants, an egg meets 128% of the choline requirement. And so thinking beyond the amino acid composition, I want to just have you bear in mind that the choline content is really important in this story. So what we've done is to look at the change in the infant plasma concentrations of a number of nutrient biomarkers. We've looked at amino acid concentrations. We focused quite a bit on the choline and choline metabolic pathways. So choline, betaine, TMAO, phosphatidylcholines, and sphingobiolin's, these are all biomarkers on the choline metabolic pathway. We've also looked at essential fatty acids and serum retinol and vitamin B12. And these analyses were done either at the Washington University Metabolomics Core Facility or at NetLab in Ecuador. So what have we seen? So first I wanted to show you the effects on amino acids. And this is the full amino acid profile for the infants in the study. And what's on the left is the effect size. So this is the concentration in the egg group minus the control group over the pooled standard deviation. So they're all on the same scale. And they're ordered by largest positive effects to the largest negative effects. And what you can see is that there were significant elevations in many amino acid concentration in the egg group. But when we corrected for multiple comparisons, so there's a lot of hypothesis testing in just this one table. So when we corrected for multiple hypothesis testing, only 3 of the amino acids kind of hung in there as marginally significantly elevated in the egg group. So not as much as we might expect given the amino acid content of the eggs. Now when we turn to choline, on the other hand, we saw that choline specifically and then all of these other nutrients on the choline metabolic pathway were largely and statistically significantly affected by the intervention. And they remained statistically significant even when we adjusted for multiple comparisons. The other nutrient that's of importance is DHA, which is a long-chain polyunsaturated fatty acid. That was significantly elevated as well in the egg group relative to the control group. Now next, we wanted to look at could any of these nutrients be explaining some of the growth effects that we saw. So we've done some mediation analyses with these data. And so as Chessa mentioned, the overall effect size that we saw on length for HC score was about 0.63. So then we looked at the nutrients in this mediation analysis, and the strongest one to come through was choline. We also saw that betaine was acting as a potential mediator, but betaine is produced from choline, and so these are basically telling us the same thing. And this explained about 15% of the effect could be attributed to choline. DHA was explaining about 4.5% of the effects of the intervention, and the amino acid histidine was explaining about 4.2% of the effects. The DHA and histidine effects were marginal on their own, but when taken together, these three nutrient biomarkers were explaining about 24% of the growth effect. So that's pretty substantial, but it also is illustrating that there's a lot that's unexplained beyond just these three nutrients. So what is choline? Choline is a conditionally essential nutrient. Requirements are higher during pregnancy and early infancy. For those of us in the room, all of us adults, we can biosynthesize sufficient amount of choline that we don't really need to get that much from our diet. But during pregnancy and early infancy, there's a need for additional choline to come in through the diet because they can't produce enough to meet the demand. It's intrinsic to the neurotransmitter acetylcholine, and it plays an important role in the development of the hippocampus, critical for declarative memory formation. It's also involved in the one-carbon metabolic cycle for DNA methylation, and this particular pathway may be important in the context of B12 deficiency, which was relatively high in our study population. Oh, I forgot to mention, we did look at B12 status and as well as serum retinol, and we did not see that there was an effect of the intervention on those two nutrients. So while B12 deficiency was a problem, the egg intervention did not affect B12 status. But when we looked at the choline effects and those one-carbon metabolic cycle biomarkers, there seemed to be the fact that choline maybe was acting in lieu of B12 on that pathway. DHA is a long-chain polyunsaturated omega-3 fatty acid. It can be either consumed directly, or it can be produced in a series of metabolic steps from the essential fatty acid alpha-linolenic acid. It's the predominant omega-3 polyunsaturated fatty acid in the brain. It's important in neurogenesis, neurotransmission, myelination, synaptic plasticity, among other functions. So while we were looking at the effects of these nutrients as a mediator for growth, we also hypothesized that these two nutrients in particular are also acting maybe as mediators for cognitive development and cognitive function. Unfortunately, as Chessa mentioned, our sample size was a bit small in this study, and so we've not been able to directly test that hypothesis in this study. But to summarize the results of the Loon study, the Loon Project, we've seen a significant effect on length for HC score and a large reduction in the prevalence of stunting. Compared to other complementary feeding interventions, this seems to be larger than the global average estimates. But if you really look, in meta-analyses, we are combining many different types of complementary feeding interventions to come up with a single point estimate. When you go into the individual studies that have seen larger effects, many of those have had targeted messages on eggs and improvements in egg consumption. So the higher effect size estimates in the literature seem to be studies that had a focus on eggs. So we think that the effects that we see in this study is biologically plausible and consistent with some of the other interventions. We also have these biomarker analyses that provide some robust evidence of important metabolic pathways that are affected, choline in particular, as well as several amino acids and DHA. Now, as a result of this study, we found, well, actually, leading up to the study, one of the reasons why infants were not consuming eggs in these study communities was because of recommendations coming through the Ministry of Health that encouraged women to withhold eggs from infants until after 1 year of age. And some of those recommendations were driven out of older evidence suggesting that early introduction of eggs might induce allergies in young children. But when we presented these results to the Ministry of Health after the conclusion of the trial, they had decided to shift their infant feeding guidelines and change their recommendations. And they now recommend introducing eggs, I think it's at 7 months, right? We were recommending 6, but they went with 7.
They never do what we want.
Yeah, but we were really thrilled about that outcome of the study, to see a change of policy as a result of this project. But we do acknowledge that this project was a small study and has some limitations. We also had an unexpectedly large effect. This was larger than we had hypothesized we might see. There were some baseline imbalance on some of the measurements, and so because of these limitations, we really think that it warrants replication in another setting. And so I'm happy to say that we're actually launching a new trial, hopefully next month, if not early in January. So the Madeira Study will be a replication trial of the Loon Project, and we'll have many of the same elements and objectives of the Loon Study, but will be done in a very different context. So the Madeira project is going to be, we're rolling this out in the Mangochi district in southern Malawi. It'll be located in the rural Lungwena communities next to Lake Malawi. And it will provide a nice contrast to the Ecuador site. In a very different type of community, a different type of setting, different background diets, different prevalence of infection and other factors. So if we can see similar benefits in this very different population, I think it will provide strong evidence of the benefits of eggs. So the objectives of the Madeira project are going to be, just as with the Loon Project, to evaluate the effects on growth and the prevalence of stunting, to explore metabolic pathways. But then we have 2 additional objectives that we're quite excited about. One is to look at the effect of eggs on gut health and the microbiome, and 4th, to look at the effects of eggs on child development. And we have tests in which we're looking at declarative memory specifically in young children using eye tracking testing. So the study design for the new project is again going to be a randomized controlled trial with 2 groups. The egg group will be given 1 egg a day for 6 months, enrolling between the ages of 6 to 9 months. Our control group will be the usual diet with a delayed intervention food basket that's given at the end of the study at 6 months. We have a larger sample size for the Malawi study, 331 children per group. And so I think this is going to allow us to explore some other things. Now, finally, we also are interested in looking beyond complementary feeding. Eggs offer an important opportunity for improving maternal nutrition as well as child nutrition. Choline in particular is important in fetal development, fetal brain development, and so we are exploring options and hoping to get some funding to be able to look at eggs during pregnancy on maternal nutrition, birth weight, and infant growth. And then we acknowledge this intervention design and these efficacy trials that we've done are not scalable. And so we would like to look at, in new studies, looking at the effectiveness of promotion of eggs, potentially through enhanced village poultry production with targeted behavior change communication or social marketing messaging, promoting eggs, to look at both production measures as well as consumption measures among women and children, and growth and development outcomes as well. So in conclusion, we are, as Chessa mentioned, we are really excited about eggs. We think that eggs offer a wonderful opportunity for improving the quality of the diets for infants. Dietary diversity, particularly among infants aged 6 to 12 months old, is really poor. It's not great among 6 to 24-month-olds, but when you narrow in on the 6 to 12-month age range, it's really quite limited. And so there's a need to try to figure out ways to introduce nutritious, nutrient-dense foods early in the complementary feeding period. And eggs are a fantastic opportunity for that. They're easier to introduce. They're feasible to provide to young children. They can be easily prepared and served to young children. They are locally available and may be a bit more affordable than other animal source foods. And so we think they offer untapped potential to address many of the salient issues in maternal and child undernutrition. And so finally, I'd like to acknowledge all of our fantastic collaborators and colleagues on this project. Individuals at Washington University in St. Louis, Universidad San Francisco de Quito, NetLab in Ecuador, other colleagues of mine at UC Davis, Chessa as well, the Mateel Institute, and new colleagues at the University of Malawi College of Medicine for the Madeira Project, as well as the community members and enthusiastic egg consumers in the community of Pasto Calle and our new communities in Lumbuena in Malawi. So, and also to just finally thank our funders, the Mateel Institute, as well as the Bill and Melinda Gates Foundation, who funded the biomarker analyses from the Loon Study and are funding the Madeira Project coming up. So with that, we're happy to take any questions that anyone might have.
Thank you so much. This is a great time to introduce our discussant. Frank Berthe is a senior livestock expert at the bank, and it can provide us with the perfect transition to discuss the implication of the funds into policy.
Thank you. Thank you very much. Thanks for the excellent presentation. I had been exposed to the Loon Project through different seminars. There was a great workshop down in Florida under the umbrella of this USAID Feed the Future Innovation Labs, bringing the livestock lab and the nutrition lab together. And I think it was great. And thank you, Era and Aaron, to put this seminar together. I think you've done a great job in putting the context, these 150 plus million children under the age of 5 and suffering stunting as a result of undernutrition. The figures are so from WHO that 45% of child death under 5 are caused by undernutrition. So we have a fairly dramatic background there. I'd like to say something there that it's not necessarily just an issue in developing countries. We have increasing concerns in developed countries, and figures recently from UK showing that in the young people and young female more particularly, we have issues of nutrient deficiency, iron, selenium, magnesium. So this is something that we have to look at very carefully. Your research is just fantastic. The figures are striking. So 47% in reduction of the prevalence of stunting, and 74% for the underweight. This is just amazing. This is a dream. And I think it comes very well to fit this corpus, this growing corpus of evidence about the contribution of animal source foods to nutrition. So this is something we know, and I think that we've known that for a long time. This is a traditional knowledge that we have in families and in communities, but bringing science into that is extremely important. It comes a bit with a good news on our side. What we see at the World Bank is that countries are increasingly investing in livestock, and so bridging the nutrition and livestock is something that is getting very on our table. Basically, what we see is that livestock and animal source foods will be essential to deliver on our objectives in nutrition. And I think that we could move the needle a bit further by saying that without livestock, we won't deliver on nutrition. And I think that your work is going into that direction. Now, what we want to make sure as a development community is that when we work on livestock, we deliver also on nutrition, and we have to make that connection. What we know is that we have to go beyond production and productivity and making sure that we deliver on nutrition. And the pathway between the two is fairly complex, because if you increase the income of the households based on chicken and eggs, you just don't know what this will be used for, and the loops back to nutrition, and especially for children, can be fairly complex. So we have to work on those pathways, better understand those pathways to make sure that we deliver. I'm a vet by training, so I won't miss an opportunity to say that we have also to put that in the broader picture of livestock. They come with risk. We could mention the competition for natural resources, but one risk that I would like to mention here is, of course, zoonotic diseases, food safety, and those issues have to be worked also along the scaling up. So I really enjoy this work. Of course, I think it leaves us with a fairly complex equation on the table: how to scale up and how to make best use of this knowledge in our projects. And I will stop there to open the discussion.
Great. Thanks a lot. Let's open up to questions, comments.
Consider the color of eggs for the cultural sensitivity of the people, white versus brown. Is there any desire by the people for any other color of egg?
No.
I would say that wasn't in any of our quality research in Ecuador. In the research we've done in Malawi, most of the eggs are brown. I don't see too many white eggs.
Right.
They were multicolored in Ecuador because it was a cooperative we bought the eggs from, that they were, a lot of different small scale producers were bringing them in. So, but probably with the small scale producers, it's more typical not to have the pure white eggs.
I just saw white eggs, that's why I was asking.
Yeah.
A mix of colors.
Or maybe I think
The one you had from Loon was multicolored.
yeah, that's what I remember. Yeah. Yeah. But it's an interesting question, and I think we should probably try to ferret that out a little bit more when we do our policy work.
Two questions for you. One, what is the nutrient profile of eggs compared to the next cheapest animal source of food? Like how much overlap is there? And the other one is, are there known rural areas or countries where eggs are definitely part of the diet, and others where eggs, especially for young children in the complementary feeding, where eggs are taboos, are definitely not, but the children where it would be a little bit understood.
What we saw in our 2014 papers, we used all the DHS data that were available to kind of look what's going on in all the different countries. Definitely in Latin America, it was higher. I think a lot of that could be an income effect. They're just by and large, as poor as some of these communities are, they were better off than some of these really, really poor communities. We looked into the literature on taboos. We didn't find a lot. You hear about it a lot. Oh, I went to this country and they said this about the egg. We found some examples, and we do report them in the 2014 paper. I think our hypothesis, the egg consumption was way up in Ecuador. And as I said, the marketing around them was just really fabulous. So hypothesis, at least, is that if you understand the cultural milieu, kind of in which you're trying to introduce eggs, and appropriately design your strategy in terms of behavioral change, communication, and marketing. You're not selling them, you are selling public health, you're selling, even when you're giving them away, it's a form of, you got to convince people. That a lot of these can be overcome. But, clearly qualitative research is you can't start anything until you do. Do you want to answer the other question about the animal source foods? Or?
Yeah. It varies by nutrient is the short answer, but I would say for some nutrients, meat like chicken or beef would be better sources, more bioavailable for B12 and iron than eggs. Eggs are not particularly bioavailable for iron and for B12. But for choline, eggs are much better. And the amino acid composition of the eggs is a bit better. It's better tailored to the infant requirements than other meats. But I honestly haven't looked at it that specifically for chicken or beef or other meat products. Now, the other thing that I think is important, both for eggs as well as dairy to a large degree, is that we should be thinking about these products not just for their nutrient composition, but for other bioactives, because eggs and milk are both designed to promote growth and development of the organism that they are suited for. And so they have growth factors intrinsic in the matrix. And those hormones or those other components of the food product may also have growth benefits on children. We didn't have the capability to test all of those. We will be looking a little bit at IGF-1, which is one growth measure in infants in Ecuador, and we haven't done that yet. So, yeah.
Just to follow up, the other thing is a very practical point, I think, which is that even if chicken and beef were much preferable, you can't get them into children 6 to 12 months old. I mean, it's just it's a basic issue around chewing, swallowing, the whole bit. So eggs, again, have this amazing plus there in terms of just practical intake.
Right.
Exactly. And then when you compare them to dairy, for instance, shelf stability of eggs is a bit better than dairy. And so they're, from a food safety perspective, they're a bit better than whole milk, dairy products. So I think there's a lot of reasons why they are really optimal for infant health, for infant nutrition.
Can I follow up on your comment that on the affordability, you said that there are areas where they could afford them and where they couldn't. Aren't most of the people with this problem very impoverished, heavily involved in heavy manual labor, at least the parents are, and they have to pay out of that manual labor? Already undernourished or malnourished. Therefore, if they're going to invest in eggs, what are they going to have to give up? And if they give it up, is that going to impact on their economic opportunities, like giving up calories when you have manual labor to do and you're already undernourished?
Well, first, we hypothesized that we're having more eggs in Latin America, the Caribbean, compared to many of the other countries we looked at, because many of these communities are better off. But even within some of the African and Asian communities, we're seeing differences of young child egg consumption. It's always higher among women. It's not super high, but there seems to be, more of an, again, to what extent is it something that kids are just, there's this belief that maybe they shouldn't be getting eggs this young. The thinking, our thinking, which is sort of the second sort of research area, is that, the overall livelihoods around poultry need to be improved. It absolutely makes sense that families sell eggs rather than eat them, because it's an income generating, they need the money. I mean, they need the income. It's not an irrational decision at all. It's a very rational decision. We certainly wouldn't say only buy eggs and don't, pay for your school fees and don't buy cooking oil and seeds to replant and things like that. That would never be the message. But the overall goal would be more economic development. And there is some really interesting work in Zambia that shows with small scale cooperative village production that with better chicken breeds, with better feed, Newcastle vaccine, that production goes up from 100 eggs a year to 300. And they also found that there's almost a threshold effect. If they sell enough eggs, then there's enough eggs to then start actually consuming them within the family. So that's, some early good data, but I think, again, all these, we just have to learn a lot more about what are the pathways through which improving egg production will impact on household livelihoods, including, the egg consumption.
Just to add one point to that, one thing that we did see that they gave up in this study area was snack foods.
Yeah.
So the eggs replaced snack foods. to some degree in the infant diets in the Loon Project. So I would see that as a very positive replacement. Snacks. So like junk food, yeah, chips and sweets and things.
Right, but they were buying the egg.
Yeah, they were being given the egg.
Right,
But
I think you were referring to a situation where family
And the trade-off between buying
eggs.
eggs versus something else that allow them to work longer, harder, and earn more money.
yeah.
That's the kind of trade-off I'm looking at. I'm sorry, I come from an agronomic crop production perspective where I find farmers with only half the calories they need. That's just
It might be useful to just one comment along this line to follow up these kids now, or just these communities where these eggs were enough. You primed them on the importance of egg consumption, and they were very sort of aware of the importance of egg consumption. And if you follow up sort of their practices without providing eggs, but just looking at their diet composition.
Yeah.
Laura, she had summer in Ecuador, looking at egg consumption, what's happening with the kids in terms of their growth. So absolutely, and we saw by the end of the study that egg consumption wasn't bad in the control group. They were the message was out there, kind of thing. It wasn't a cluster randomized design. We couldn't do that. So, but you're absolutely right. And I think there will be some long-term involvement in that community, as well as in another community, actually much poorer community, where we would have liked to have done the study because much higher rates of baseline rates of stunting. We just weren't able to because again, it's a very small budget. They're looking. Laura, the students are looking at livelihoods. They're looking at what's going on in terms of chicken production, and so really trying to get a grasp on some of these agricultural nutrition links. Yeah. Because the economics ultimately is what's going to drive.
Aaron has a few questions from the online community.
Thank you. Yeah, we actually have quite a few. I'm going to run through roughly half of them, so maybe take some notes. So the first from Christina Jane Granger with the Spring Project is, in the new project, there's no scope or bandwidth to trial different doses, like less than once per day. So is it possible that there could be a cohort doing something like 3 to 4 times a week as opposed to every day each week in terms of egg consumption? Next, a brief question from Diana on whether you're including any effects or saw any effects on infection rates during the interventions. And then from Deirdre, she has a series. I'm just going to put a couple of them on the table. One just in terms of intervention details, just to confirm the eggs were distributed at no cost. And then in terms of kind of follow-up, have the mothers or households, are they still sort of accessing eggs either through home production or purchase or anything like that? And then a question on, was there a lot of recognition of the importance for eggs based on the social marketing that were done? And I don't know, Frank, maybe from the livestock perspective, there might be sort of notes on the social marketing for consumption on that end. Thanks.
Yeah, so the first question was about whether we have the bandwidth to test basically dose response in the new project. Unfortunately, no. The primary objective for that trial has been to do a replication of the Loon Project, and so we're doing the same dose, which is 1 egg per day, and so we won't really be able to, within that sample size and within this particular trial, look at dose response. We've also been very strongly encouraged by the Gates Foundation to provide the results quickly, to be able to get an answer quickly as to whether there is generalizability across different settings. And so the focus has really been to try to keep it simple and just focus on the same that we had done in Ecuador. The infection rates in the loon, we did look at those, and they're published in the pediatrics paper. We saw no effects on respiratory infections. We did see a little bit of an increase in the prevalence of diarrhea, which was a little surprising. And we're not quite sure what to make of that. There is a perception that eggs are associated with an increase with a risk of stomach upset in young children, and because this is an unblinded intervention, that may be explaining it. It could be, though, a real thing, and we're not quite sure what to make of that. The eggs were provided at no cost, so that was one of the questions. Were we able to provide the eggs at no cost? Yes, that they were delivered weekly by a local woman who distributed all the eggs and kept track of how many of the eggs were consumed, or she asked questions about how many of the eggs were consumed in the past week. And then the question, are mothers still providing the eggs? I think Laura Iannotti will be looking at that in the follow-up study that she's been doing. I haven't seen those results yet, so I'm not sure. Is there anything I missed?
I think that the recognition of eggs is beneficial. Yeah, there's some pretty neat qualitative research, and that paper is under review right now. But what the mothers are saying, whose kids got the eggs, it was unblinded, about how they were acting and how they, just, they sense now, unblinded, but we have some really, really wonderful quotes from these mothers about how much they thought that these eggs were helping their kids. And just to talk a bit about the infection, they just presented the SHINE results last week. This was huge, water sanitation. They, latrines, they dug wells. It was just an amazing effort. And they also found an increase in diarrhea in their intervention group. And that just doesn't make sense. And so I think that, the hypothesis, certainly in Ecuador, is that you want to do something new. And, there's just, the mothers are maybe just more attentive, like, just sort of making, my kid is getting eggs, for the first time, so that's why, maybe. And so I think the hypothesis is more just a kind of response bias than in fact, but it will be looked at in the Madeira Project carefully. So.
And when does, in terms of the eggs, the eggs basically are the babies that we have. So the nutrition quality of the egg is linked to the nutrition quality of the chicken that lays it. I wonder how many eggs you tested to see what the variation of the nutrition profile was across the eggs. And because it brings me on to how you scale it up. Because in Bangladesh, there is a lot of backyard poultry. And in fact, I think HKI saw better results on anthropometric status when they introduced chickens and things in their projects after a while. But it was interesting because the Department of Health decided to collaborate with the Department of Agriculture and, essentially introduce better chickens. So they bred better chickens and they released these chickens to the households. And it was in a bank project. It was failing quite badly because when they went back a little later, all the households had given up on the new chickens and were all back to the old chickens. And so I actually was moved to Bangladesh to try and sort out this link of this ag nutrition project. And the ag guys were like, well, it's the household's fault. We gave them better chickens. They are better chickens. They lay more. The households agree they lay more, and they still don't raise them. And I'm like, households aren't usually stupid. So I went out to the field and started talking to the women to say, so why did you give up these chickens? And this one woman in the end said to me, she said, well, the chickens were stupid. And I was sitting there thinking, who knew chickens went to university? Didn't know chickens were, how can you have stupid chickens? And I said, well, what do you mean your chickens were stupid? Why didn't you do them? And she said, well, because the normal backyard poultry grows up and is used to the fact of what will eat it and what won't eat it, and therefore it knows where to run and hide. And these new modern chickens that have been obviously bred in the more factory sense, she didn't have a clue, and she said they just stood there and they just got eaten by everything. So she says, why we have the chicken, it laid more, but it just didn't live very long. It was like the old chicken. Yeah. So it was a good learner for me in the sense of, okay, this chicken rearing is not quite as simple as I thought it was. But it's also, there's, I think, a nutritional difference in the quality of an egg from a foraging, basically semi-wild chicken versus the chickens that you do. And I'm wondering how you're factoring that in and whether
there may be something in a comparison between Malawi and Ecuador in terms of the nutrition quality of the eggs.
Yeah. So we collected a small sample of eggs in Ecuador. It was 10 eggs that we had collected. So and then the nutrient composition analysis was done on a homogenized batch of the eggs, because to do a full nutrient profile on the eggs is quite expensive, and we didn't have the resources to do egg by egg that would give you a estimate of variability. So all we have is the average composition for the eggs that were measured. We are planning to collect eggs, and we have the funding to collect eggs in Malawi. So we'll be able to compare the Malawian eggs to the Ecuadorian eggs. The Malawian eggs are going to be from a large-scale producer, because we needed to have thousands of eggs a week in order to meet the demand of the project, and we wanted something that was a reliable and consistent supply. So we're buying from a large supplier, but they've agreed to meet with us, and we'll collect eggs from them. We'll also be collecting data on their feed practices and what they're doing to feed the chickens and raise the chickens, so we'll have a bit more data on that. And then we also hope to do is collect eggs in the communities to look at the local smallholder farmer eggs, the ones that are foraging in the community. So we'll have some comparison of the village eggs versus the large-scale producer eggs, and have some comparison there. Again, I don't know that we're going to have, I think we might still need to do single batch sampling because of the cost of it. So we won't have standard deviations on these things, but just kind of qualitative, the mean of this versus that and that. So to sort of descriptive wise, see what the quality is.
Yeah, and getting to your question about what you observed, this HKI project. We're really doing a lot of interacting now with a lot of researchers and practitioners that are trying to do these small scale projects. And really what they're looking at is coops, appropriate technology, small coops, and there's a couple reasons for that. Exactly what you said. These chickens aren't bred to run away from these predators, and so they need to be protected. But another really key reason, if you remember that picture I showed earlier on, chicken droppings are full of bad stuff for kids. You're talking E. coli, Campylobacter, and you think of, so there's this real trade-off. You don't, that on the one hand, eggs are very nutritious, but on the other hand, there's this infection-nutrition interaction that really drives growth. And so there's a need to, protect the outside world from pathogens carried by chickens, as well as to protect the chickens from those things that want to, the challenge the challenge you have at that point is you need a supply chain. You need to get food to those breeders, because they can't just live on the stuff that the little, foraging chickens can. So, it solves certain problems. It raises other potential problems that need to be addressed. But there's some really, there's more and more examples. There's, some work in Zambia, there's work in Ghana, really trying to look at this. Because ultimately, I think, we have to, that said, DHA is much higher usually in the foraging chickens. DHA is expensive. That's why when you go to supermarkets here, you pay a lot more for DHA enhanced eggs. So that's not going to be part of the what's going to go on if, in these smaller coops.
Unfortunately, we ran out of time. It's already 2 o'clock. It would have gone on forever. There are additional questions. At 2:30? Yeah. Perfect. Yeah. Okay, so there's plenty. We had 2 questions over there. We can go on. Okay, I'll go around. And we start with this question.
I just had a follow-up actually to the diarrhea, and just wondered if the caregivers were given education on hygiene and food safety when preparing the eggs, fully cooking or washing the hands.
Yeah, we definitely talked about in qualitative work around how they cook the eggs, because we really wanted to be sure that they were cooked in a way that you don't want to give a hard-boiled egg to a 6-month-old. That's just really a bad idea. So there were some basic messages around hygiene, as I recall, but we weren't doing any observations around that. So I can't directly, but I think, yeah, messages around, certainly around egg preparation, egg storage, and yeah.
Yeah, and in the Malawi project, there'll be targeted messages about complementary food safety, food hygiene for both groups. The egg group will get some specific messages about egg handling and preparation, but both groups will get food hygiene messages. Yeah.
I may have missed it, but I wanted to understand how you calculated this change in stunting. So over what period of time? When did you initially reflect the stunting rate? And when you say it was, 47%, so how many percentage points? And that's sort of an average reduction amongst those measured, or can you clarify a little bit how you deal with that?
Sure. So it was a 6-month intervention, and the children were 6 to 9 months when they entered the study. So they were 12 to 15 months when they ended the study.
So it's a growth alternate rate or actual stunting? Stunting usually takes a while to show up.
Right. We measured stunting initially, and we measured length for age initially. We measured length for age at the end of the study, so we could calculate changes in stunting. And we also used a difference of difference analysis.
We did. We used GLM models to look at the prevalence of stunting in the intervention versus control group at Endline, adjusting for the baseline differences. So it was basically the cross-sectional adjusted for the baseline was the primary analysis.
And so, 12 percentage points? How many actual
Percentage points.
Look it up.
Yeah, I'd have to look at weeks. Actually, you know what? It's in here.
In the table.
Yeah.
Yeah.
I have to jump back all the way. Okay, so this is the end point over here. So this is the egg group was 21% versus 29%. But as I mentioned, there was baseline difference in some imbalance in the stunting prevalence in the two groups. So the stunting difference really became apparent when we adjusted for that baseline difference.
So stunting was actually 26% at baseline and 21% at baseline?
26% versus 29% baseline to Endline and control group. And then egg group was 37, sorry, 32%. The numbers in parentheses were the percent. 32% versus 40% in the control group. And then the egg group was 47% at baseline and 28% at Endline. So there was a stunting reduction, actually, not just a difference between the groups. Yeah.
When you're adding that new measure on early developmental course, I'm wondering if you've thought about the idea of just occurs to me that you try feeding eggs to a baby, and it takes a lot of interaction, and I'm wondering if that feeding of the egg is actually going to provide information.
That's a great question. Okay, so to repeat the question, it was basically to paraphrase. When a caregiver is providing the egg to a child, there's a fair bit of interaction and encouragement to get them to eat the egg, and so you're wondering if we were going to be able, if that might have an effect on child development and how would we address it. I think it's a great question, and we hadn't thought about that specifically. So you raised a really good point that we should be thinking about. We will be having some observed feeding in the trial. That's somewhat of an addition compared to the Loon Project. We'll be having observed feeding on a couple of days a week. So we would have the opportunity within the observations to be able to look at responsive feeding or caregiver-child interaction, possibly. Yeah. Yes.
Taylor Wallace, a researcher with George Mason University, and I've been doing research now for quite some time for quite
That's a
some
great
time.
comment.
So, I wanted to just make 2 points, moving into the Malawi study, because actually, as you know, choline's only been recognized in the U.S. as an essential nutrient since 1998. We actually don't have a lot of data, even in the developed world, on choline. I would argue some of the best data that we do have are rat studies out of Steve Ziezel's lab from the late 1990s. And there's some real critical windows during pregnancy when you look at neuronal development in the rat and long-term outcomes as the rat ages, which I think is really interesting. The other thing that I think might be really, really interesting, and it kind of brings back to your point on animal nutrition, is that in the U.S., at least for decades, since the 40s maybe, we've been supplementing dairy cattle with choline to prevent fatty liver disease. When you are deficient in choline, you develop non-alcoholic fatty liver. And we know in women, in humans, that serum choline or breast choline is about 10 times more concentrated in choline versus serum levels, because it's, like you said, very important for neuronal development. I think it would be really neat in the study to look at the temporary instance of fatty liver in these women in Malawi. That might be a really neat measure that you could do relatively inexpensively. And it would provide the public health community in general with some really good data.
Yeah, great point.
Big fan.
Great, no, great point. Yeah, we were definitely aware of the work that Ziezel's doing on with the rats, and that's why one of the reasons I think, again, we were so kind of focused in on the study and on eggs and choline in particular. And I think your point, I didn't know about the fatty liver at all in women. So if
Well,
it's
this is just kind of a thing going on in my head with the dairy cattle and the serum versus breast.
these are one
two similar mechanisms. It could happen.
Well, we really want to do the pregnancy study as another proof of concept.
Yeah.
And so, if we end up managing to find some funding for this, then we'll have to collaborate.
Well, and I've been wanting, if we were to do a maternal nutrition study, to look at breast milk composition and choline to see if that was improved with the intervention.
Well, we would expect it to be, but we'd have to measure it.
It's all about funding, but I've actually wanted to go to fertility clinics and get women before they actually get artificially inseminated and manage their livers and look at liver function throughout lactation. So we've got a lot of really deep areas.
That's not on our research.
But we'll look forward to your paper.
There are some more questions online.
Yeah.
Okay,
Thank you. Yeah, we still have a bunch coming in. I'll just go through 2 or 3 here. So one was from Colleen. It's sort of a note on the research, just to ask whether energy consumption was included in your regression model when you were running your numbers. And from Patrick, asked a couple questions. He followed up with one that, since we're talking about stunting, is important. Can we get confirmation that the maximum age that stunting interventions or what's the maximum age where stunting interventions may be able to cure or resolve stunting? From my reading, it might be around 2 years old. So just looking for some confirmation on that. And then where did it go? From Bindi Borg, a question on what might be the impacts of egg consumption on adolescent girls and pregnant women in terms of child stunting and health outcomes. Thanks.
Okay, so let me just take the first crack. That's the great thing about eggs, because we always make all these jokes. So energy consumption. So no, because we did not have the funding to do quantitative 24-hour recalls. What we did is we had 14 kind of foods that we asked around, and that's how we were able to find this difference, as Kristen mentioned, in consumption of sugary foods. But we could not quantitatively assess, so we did not do that.
Can you mention in Malawi?
We are going to do it in Malawi. Yeah, we have, it's going to be really a lot of dietary data. There's going to be duplicate surveys, there's baseline, midline, and end-line surveys. So we're going to have, all the quantitative, the data to look specifically at that question. The max age for stunting reduction. I love that question. Because, we talk about the 1,000-day window. And, I purposely didn't put the 1,000-day window in my slide because I frankly think the 3 to 5-year-old age range is terribly neglected. We talk about school nutrition, but we talk about under twos, but it's like kids still have to get from 2 to 5. And so there's no question that maximum growth is in kind of the first 2 years. I mean, it starts, if you look at different trajectory, they're growing, but they're slowing down in their growth. And so there's a lot of reasons for intervening early, and that's particularly why we wanted to get these kids really young. In fact, we wanted 6 to 8 months, and we just couldn't quite recruit enough kids in our narrow time frame to get the kids, so we had to expand the recruitment criteria.
Can I add something
Sure.
to that? Just to add about adolescence as well. I mean, I think that there are growth spurts that are occurring in adolescence, and there is the potential to affect growth in adolescent girls in particular. No nutritional interventions have seen effects, but certainly things like delaying pregnancy and delaying marriage in populations where there's early age at marriage, that can have an effect on adult female height, and that has an effect on all sorts of things for women, reproductive wise. So I think there's opportunities to improve growth for a wider period than just the first 2 years.
Absolutely.
Yeah. And then what was the third one?
That was a question. That was the third one.
Oh, okay.
Impact on eggs in adolescent girls. It'd be great to study it, but we don't know. But adolescent girls, there's so much interest right now. There's just a lot of Spring just did a 2-day meeting on adolescent growth, and there's just a groundswell, I would say, on interest right now on adolescent girls, and so it would be interesting to look. And we don't know very much about what they're eating. And the one systematic review that Spring commissioned from Zufi Bhutta and his group, the conclusion was we have a lot of evidence that we have no evidence about what they're eating. And I thought, priceless, because it's we don't know. DHS doesn't collect it. We don't have any large national surveys that are representative globally that would give us that kind of information.
Yeah. Two more questions.
Yes. So I just had a question. Based on these results that you're showing, I'm wondering if you looked at any interaction effects with something at baseline, and maybe I missed it. So, where those kids were already stunted, did they benefit more? And the differences between the control and the intervention group would maybe point towards that, given the differences that you see in those two groups. And then did you do anything by age? And again, I know that for age, the sample size is maybe not big enough to detect difference between, like, if you introduce a 6-month-old versus 7 months versus 8 months versus 9 or... Anyway, so I'm just wondering if you looked at differential impact of the intervention based on nutritional status at baseline and age and enrollment.
Laura did most of the analyses, but I don't think that I'm aware at all that we look by age. Again, the sample size we were at the limit for power to look at growth. I mean, we based our sample size on B12, and we didn't find an effect in B12, but that's how we based our sample size. Because again, this was all driven by budget. It was really, we were so grateful to Mateo, but that said, it was, nobody took salary, even Will down in Ecuador. I mean, we wanted to do this so badly that, we paid field workers and we paid lab costs, and that was it.
So in fact, I mean, we weren't really very powered to look at interactions. We did do some exploratory analysis, not with age, but we were looking at, given these numbers, we were looking at recovery from stunting, which we didn't really think was possible, but we saw some evidence of that in this cohort, that in the egg group there was some evidence of recovery from stunting, that because the whole distribution was being shifted up.
Totally
so,
moved.
Yeah, but we did not look at interactions with the way that you're looking at it.
in the model, so.
Right,
Yeah.
baseline we did.
Yeah.
Yeah.
I mean, but how did you ensure in Ecuador and how will you ensure in Malawi that child is the entire egg? Because it's actually hard to get a young child to eat an entire egg.
Yeah.
Yeah.
Especially when you mash it up and you blend it with breast milk or
Right.
water or something.
Right, yeah. Right. So egg consumption, we measured just by, did the child eat an egg yesterday? And we didn't even measure portion. A little, I mean, it was, did
In
the
the
child,
qualitative research we did.
yeah, in the qualitative we did, but that was, yeah. What we found, at least at the end line, 80% of the mothers in the control group, in the intervention group reported that the child had received egg in the previous day. So I think that's something we can look at with the observation in Malawi 2 days a week. We can look at that much more carefully.
Yeah, and
Yeah.
and in fact, in the qualitative research, some of the mothers were saying that when they were young, they weren't eating the whole egg.
Yeah.
The mother would prepare the egg, the child would eat however much they would eat, and then they would share the remainders. But what they did say, and this was again in the qualitative research, was that as the child got older, they were consuming more of the egg, and that by the end of the study, they were consuming the whole egg themselves.
Right.
Which is, you would expect for a child, developmentally for a child, that they're more able to eat a whole egg and their appetite would be larger to eat the whole egg at that point. In the Malawi project, we will be collecting more data we'll have the observations, but we'll also have reported portion size consumed, and we're working on pictographs to help capture that information a little bit better.
Yeah, and actually, Anne, could you get some pictures of you feeding eggs to your grandchild? I'm very curious because, if anybody, hey. We got, I have hundreds of pictures of children eating, from work from a contest we did on, show us the greatest picture of your child eating. Just get the pictures because we didn't have any. And, we didn't get any of eggs. And so I just, I felt like when we're putting this together, that's something that I really do want to. Yeah. Keep it in mind. Well, aren't you convinced that your boy or girl should get an egg? And you too. There's some other day.
When we look at nutrition levels in people, there's some catchwords that are throwing up: nutrition, density, and not only your nutrients, but the minerals.
Yeah.
I think minerals get a good play in the role that they are in cellular development. So there's significant values there that need to be. And then the traceability of those minerals and nutrients back to the raw source of the feed to the chicken. And then when you get into sustainable agriculture definitions and the complexity of agriculture industries for small-scale farmers, the interplay between chickens and the small coops and beef production and how they feed off of the cow dung and the leftover grains from the cow and the sourcing them. So there's a lot of interplay back and forth, and it would be interesting, the commercial producer versus the small scale producer and what they're actually feeding the chickens, and if it's a natural diet from whatever is growing there versus they're actually supplementing the diet. So to actually do traceability in that. There's a tool online for Sustainable Development Goals, the ISDGS. ISDGS.
DGS.
Yeah, ISDGS.
Okay.
So it will help relate what you're doing to the Sustainable Development Goals to eventually affect policy. Because you're having an impact on not only nutrition and stunting, but you're also having impacts on the chicken industry, the poultry feed industry, dairy industry. And so there's a lot of industry stuff that you could potentially have an impact on in your studies.
Yeah, those are really great ideas. And if you go back to the picture of the egg that I kind of in the earlier part of the presentation, there's selenium more than 50%.
Actually from the
Copper is a big one too.
Copper,
Yeah. The selenium is really good.
For
in
copper,
the-
for brain, iron, for
right,
lungs.
exactly, yeah.
Yeah.
And so I'd say we completely agree with you, and we did not have the funding to do serum mineral analyses in the Loon study, but we are planning to do trace minerals, do a full profile, zinc, copper, selenium, iron in the infant serum samples.
Yeah. I mean, there's so many unexplored questions, both from an efficacy perspective, as well as, moving into, okay, how do we scale this, and we're nutritionists, so, obviously as we move forward, we need specs, and we need to have economists, and
Well, as a source of funding, if you're going to have a play on, what you're looking at is the ultimate outcome. And what you need to look at is how do you get to that outcome, and it's the raw material that goes into the chicken that's going to determine the quality of the egg that comes out.
yeah.
And so the chicken feed industry, the people who supply that chicken feed, those are the people that may be able to be approached for funding.
Oh, yeah.
Because they will determine the nutrient density of what's in the egg.
Yeah. Thank you.
So as I looked at this chart, I realized we didn't talk about one nutrient like in eggs, and that's lutein. And eggs are interesting because the lutein is about 3 times more bioavailable than from spinach or your green leafies. One of the things I think is really interesting is that you have a good marker for lutein status because it builds up in the macular region of the eye, and that is a very good biomarker of brain lutein status. And so you can use flicker photometry, which is an easy, simple technique to really look at cognitive function. A lot of in the developed world, macular pigmentation has really been related to cognitive outcomes. So as that crossed my mind for your second study, I don't know if you looked at lutein levels in the first study, but it might be a really interesting thing to look at because it seems like lutein got some really beneficial effects on cognition. And it's an easy
That's
biomarker
great.
to measure.
So wonderful. We have to talk to you a little bit more. No, card really is.
Yeah, that's a comment from an economist perspective. There's a lot of work done at the bank in the data group. There is the LSMF is also a Gates-funded project on integrated household surveys, and they have livelihoods, they have consumption, they have anthropometrics and livestock. There's a lot of information on agriculture, non-agricultural sources, and there's a well-developed panel also in Malawi that has been the new way to do this. And so in terms of question of development, there's a lot that you could sort of borrow from, and there is also summary meta-analysis on the interplay between livestock and food and anthropometrics and nutrition. I think it's input policy that might be useful for you to.
It's already published.
Yes.
Okay.
To look at. And it also might bring back sort of the question of measuring, especially in a low-income environment, the behavioral responses of the household. If sort of the child is consuming an egg, there might be a sort of substitution effect for adults or other members of the household. And so only with an integrated survey you can capture the behavioral responses instead of just focus on the child's outcomes. And that's a great source of questions that are already been validated.
Great.
Thank you. Very good. This has been a great presentation and discussion. Thank you everybody for coming, and thank you for organizing this.
Yeah, really.
Yeah, well, thank you
I think
so much for having us.
emails between us.
Yeah.
Thank you, Erin. Yeah, really, really great. Yeah. Thank you. Thank you.
Yeah, thank you.